Hello there and welcome to this A-Level Biology Mind tutorial on calculating genetic diversity. So today we have two key learning objectives, firstly to understand comparative genomics and secondly to understand quantitative analysis. So we've already talked about comparative genomics previously in tutorials on comparing different populations and looking at phylogenetics. So let's talk in a little bit more detail about this. So comparative genomics enables the understanding of the diversity of genes within a population or community. It also shows how these genes change over time in order to understand evolution and evolutionary relationships. So how do we measure characteristics? Well, there are some observable characteristics such as facial features, body shape and bone structure, and there are measurable characteristics, so weight, height, blood pressure, for example. So we can deduce similarity between organisms by looking at how they are different or similar in terms of observing them, so looking at their facial features, body shape, bone structure, but we can also look at measurable characteristics and see if they are similar or not in that way. Now, new techniques enable rapid sequencing of whole genomes. So for example, next generation sequencing. So by comparing the DNA sequences between species, we can determine the relationship between these organisms. So the higher the similarity between the species, the higher the similarity between the DNA sequences. Okay, so by aligning the DNA of one species to another, we can determine how similar the base DNA sequence is. So for example, we can see here that these sections of DNA are very, very similar between humans and chimpanzees, which makes sense because they're very closely related. And again, we know that by looking at this phylogenetic tree where we can see that they branch off at a point very close to each other. Whereas, for example, let's contrast human and orangutans, they branch off over here, which is a point much further away than humans and chimpanzees, which implies that they're less closely related than humans and chimpanzees. And actually, yes, if we look at their DNA sequences, they have much more differences between them. So this is how we can use DNA sequences to determine how similar organisms are. So we can compare two populations and see similar genes which could hint at a common ancestor. Now, exon sequences, which remember are the coding part of the DNA sequence, are similar between organisms. Now, the underlying base DNA sequences of different genes may be different, making DNA comparison slightly ineffective. But because mRNA contains only exons, we can compare mRNA rather than comparing DNA, because remember DNA contains exons and introns, whereas mRNA has been spliced. Those introns have been removed, which we talk about in our tutorial on transcription. So if you've forgotten that, go back and have a look. So because mRNA has only exons, it is similar to compare alignments of mRNA to see the similarity between organisms. So there are still limitations in mRNA comparison. So mRNA sequences between different organisms for the same gene can be quite different due to the degeneracy of the code, which we will have a look at on a later slide. I'll explain that word later on. Now, we can also look at similarities between organisms by looking at similarities between amino acid sequences. So amino acid sequences vary less between organisms despite the degeneracy of the genetic code. We can compare the function of proteins in different organisms, and when making evolutionary relationships, the conservation of the function of a protein in one species to another needs to be determined. So this must require structure conservation too. Now, comparative alignments of amino acid sequences of proteins from different species are the best tool. So let's have a think about what this degenerate nature of our genetic code is. So all this means, sorry, that's not very legible, is it? The degenerate nature of our genetic code. So all this means is that multiple different codons or DNA triplets code for the same amino acid, which is really beneficial because let's say there's a mutation in this thymine and that changes it to a cytosine base, it's still going to code for the same amino acid. So there's a really good benefit in the degenerate nature of the genetic code. Now, for questions on DNA similarity, they will often give you the base sequence as a diagram or a percentage similarity. Now, the higher the similarity, the more closely related the organism, but make sure to mention that the limitation of DNA comparison is the fact that the code is degenerate. So make sure to mention this in your exams if you're talking about how similar organisms are based on their DNA sequence. Now let's have a look at some quantitative analysis. So remember, quantitative just means getting numbers to analyse different variables. So scientists use sampling to study populations. So sampling needs to be random in order to minimise bias. So, for example, you can split a field into a grid and use an online calculator to randomly generate coordinates. And then you can plot those coordinates on the grid and sample. So let's say your coordinates come out as this point, and this point. Then you're going to sample in these squares. So this is a way to sample randomly. You split the field into a grid and use an online calculator to generate random coordinates. Now, we want to sample as much as possible because the larger the sample size, the more reliable the data, the more significant the data that is yielded. So in order to obtain accurate results from your investigation, sampling techniques should be tailored to the organisms that you are investigating. So for example, you could use something called a Tillgren funnel, which you might come across in your biology lessons, to sample small insects living in soil. Now, if there is a large variety in the way a species is distributed in a habitat, sometimes you may need to take a non-random sample. But if the species is equally distributed across the habitat, then it is fine to take a random sample. Now, the reason why you might want to take a non-random sample, for example, is if, for example, you want to sample woodlice in a field. Now, you're not going to find many woodlice just out in the grass, because where do they tend to be? They tend to be under rocks. So if you do this and plot a grid and generate random coordinates and none of your coordinates end up being on a rock, you might end up finding that there are no woodlice in the field, but actually they're all just under the rocks or under the logs. So this is a case where potentially you might need to take a non-random sample and make sure you sample under those logs to make sure that you include the woodlice in your data. Okay, so we often plot continuous variation on something called a normal distribution. So a normal distribution graph allows us to visualize how distributed our data points are. So these are our data points here, and you can see the spread of our data points. So normal distribution have most values closest to the mean, which is the center point. Now this is the peak on the graph here, so this is our mean. So remember, mean is the total of all values over the number of values. That's how we calculate our mean. So our mean is our total of all values divided by the number of values. Now, our standard deviation is a measure of how spread out the data is around the mean. Okay, so the standard deviation is a measure of the spread of our data around this mean. So when drawing conclusions, we need to measure how significant the difference is between samples. So in most cases, we compare the means of our samples to see how different they are. The larger the standard deviation, or for example, in this graph here, it's showing such a large standard deviation. We know that because look at that spread around the mean. Whereas this normal distribution curve shows a smaller standard deviation because this is the mean, and look at that spread around the mean. It's much smaller than our other curve here. So a larger standard deviation, or the further away values tend to be from the mean, the less confident we can be about our results, because it shows that there's more spread around the mean. So we want a smaller standard deviation to be more sure about our results. Okay, so lots of terms covered there again in this tutorial. Well done for following through to the end, and I will see you for the next one.